Hey, I thought I'd walk you through DNA replication. Basically, what it is copying the genetic code. Now, there's a couple of very good reasons why our DNAs need to be copied. Every time our cells undergo division, meaning one parent cell becomes two daughter cells, they need to make sure that their genetic material is copied completely and accurately, and that copying happens during interphase. Once the cell enters the mitotic phase, the DNA is then equally divided into two identical cells. We're looking at the mitotic phase of a diploid. Let's assume this to be a diploid organism, meaning it has two chromosomes. Now, if you look at the chromosomes, you'll notice that it is in the form of sister chromatids, meaning here's a chromosome and a replicated version of it, or an identical copy of it. Here's another chromosome with this identical copy attached at the centromere. In anaphase, the spindle fibers will attach to the centromeres and pull apart the chromosomes into two identical cells. Notice that the number of chromosomes are conserved here, meaning if the parent had one and two chromosomes to begin with, the daughter cells also have two chromosomes each once they have completed cytokinesis. In its condensed form, DNA is known as a chromosome. Chromosome is basically inactive DNA, or DNA that is not being replicated or copied. Because the DNA is folded over and over into maybe 1,000 time compaction, it is in its chromosomal stage, it is basically unable to be copied. It is it does not the information in the DNA is not readily available. So walking you through, this is what a DNA would look like when it's about to be copied. It's very all the bases and all the sequencing in it is easily readable. But once the DNA is no longer in interphase, the same DNA has to be folded and packed away so it can be easily sorted. The first compaction happens around histone molecules, which are proteins, and with the help of linker DNAs, they form something called, or form something that looks like beads. And those beads compact DNA from 2 nanometer width to about a width of 11 nanometers. Further packing results in something called an array, and they happen around specialized histone molecules known as H1A, H2A, H3, and H4. And there's a couple of pairs of each. So this one has two pairs of H1A. This one has a couple of pairs of H2A histones. This has a couple of pairs of H3 histones, and this has a couple of pairs of H4 histones. At this point, the DNA has a width of 30 nanometers. They're further compacted by radial looping around a protein scaffolding, which then grow in molecular forces, intermolecular forces, until they are compacted to a width of about 1400 nanometers, and at this point it is known as a chromosome. And it is ready to divide. It's all neatly packed and sorted, and division is easier. I want you to imagine DNA is in its uncompacted form, which is a double helical structure, basically a twisted ladder. If you can somehow imagine an untwisting of this ladder, it results in something that looks like a 2D version of the same. And if you can imagine these to be the backs of your ladder, then the nitrogenous bases kind of form the rungs of the ladder. Now, you will notice that one of the strand goes in a 5' to 3' direction, and the complementary or the anti-parallel strand goes in a 3' to 5' direction. A way to tell whether the strand is a 5' it's going in a 5' to 3' direction is by looking at the ribose molecule. Notice that the oxygen is oriented downwards in the 5' end, and on the 3' end the ribose oxygen is pointed upwards. Each ribose is attached to a phosphate and a nitrogenous base, which is complementary to the nucleotide in the 3' and notice that all the nitrogenous bases are complementary, meaning if the 5' end has a cytosine, the 3' end would have a guanine, and if the 5' end has an adenine, the 3' end would have a thymine, and so on. Thymine always goes with adenine, and cytosine always goes with guanine. Also notice that the phosphate and the nitrogenous bases are held by solid lines, meaning these are strong bonds and these do not break apart. But the nitrogenous bases are held by dotted lines. In essence, what it basically denotes is hydrogen bonding, and hydrogen bonding is more of an attractive force than strong chemical bonding. And it is necessary to in the process of DNA replication. Talk about intelligent design. Once a DNA is ready to be copied, these nitrogenous bases gets cleaved or unzipped, creating two templates which can then be replicated. Notice that an adenine and thymine have 2 hydrogen bonds, and guanine and cytosine are held by 3 hydrogen bonds. Here is a 3' to 5' strand, and when they make complementary bases, they will always be complementary in nature. For example, if this is guanine, it will always be connected to a cytosine, and the adenine will always be attached to a thymine. Now, if you recall Messelson and Stahl's semi-conservative model, each DNA strand that gets copied has or retains one parental strand around which a new daughter strand forms. And you can see that happen on the other strand as well. So here's the parental strand, and around it a new daughter strand with complementary base pairing has formed. So at the end of it, one DNA molecule would have replicated into 2 DNA molecules or DNA macromolecules, each one conserving one parental or one original strand and one newly synthesized strand. The next lesson we'll talk about DNA replication, particularly about the molecular events of DNA replication. Hopefully this helps. Thank you.